All right. I think I can see Marcus's screen. Marcus Asher is the Quail Program Coordinator for Arkansas Game and Fish Commission. He's got a bachelor's degree in wildlife ecology and management from Arkansas State University. Oh, and he's got over 10 years of experience in wildlife management and ecology. So I'm really anxious to hear what he has to tell us. Marcus?
All right, thank you, Jack. As he said, I'm the Quail Program Coordinator for Arkansas Game and Fish Commission. I've been that for about 4 years now. Previous to that, I actually worked in private lands, helping landowners, implement habitat restoration. So anyway, so we'll delve right into my talk. As everybody's probably very, very aware of and very familiar with, quail are a species that's in decline. That's not unlike most grassland and early successional species. You can see here, I have two graphs. The one on the left is the breeding bird survey data for just Arkansas. It shows that same decline that you see in the United States, which is on the right-hand side. It is of note that in the United States, it looks like we're actually seeing a little bit of an uptick overall in some of the, or a lot of the routes that are run in the US. And as we'll see here in just a minute, that's very similar to what we're seeing in some of our routes that we run for our quail call count routes in the state. So this is what I'm talking about. We kind of over the last 16 years that I had electronic data for when I came to the agency, you do see an overall decline, although here in the last year, since 2019, we are experiencing an uptick. This is not uncommon. I mean, we see ups and downs with the quail population because they are what's labeled a boom or bust species, depending on weather and conditions, habitat conditions, and so forth. But in 2020, I did want to say this, we did reduce the number of routes that were run in 2020 because over the last probably 10 years or so, staff were seeing that a lot of our routes that were run produced zeros. There was no changes in habitat. It was still some of the same old, either closed canopy forests, ag fields from border to border, no habitat there. So I didn't want to continue to essentially beat a dead horse. We know that if there's no habitat there and no habitat anywhere around, that obviously we're not going to see quail there. So we wanted to focus more on sites where there's still some habitat available and kind of monitor the population from there. Maybe even in the future, pick up a few more as we need. Overall, there was an 18% increase from 2019 to 20. We saw the largest increases in the Gulf coastal plains, but also the Ozarks and Arkansas River Valley were up as well. Mississippi Alluvial Valley and the Ouachita's decreased from 24 and 38% respectively. This is a shot of those quail call count routes that we ran, or we have ran. The green dots on the map show locations where increases were experienced, or routes where increases were experienced. Red dots show decreases, and then black is neutral. The actual size of the dot on here shows the magnitude of increase or decrease. So you can see areas such as over by Fort Chaffee Military Base, where we have 4 different routes that are run on that large acreage. That's where we have a lot of, still a good amount of quail within that area. Also, a lot of the areas within the north central Ozarks, and then some, down in the Gulf coastal plains are still showing some decent numbers of quail. So those previous quail call count routes were basically, for the most part, randomly selected within the landscape. They weren't necessarily drawn to any particular WMA, although some were, just like in that case in Fort Chaffee. So they may or may not be showing a whole lot of what our management practices are, in, you know, how they are improving our quail populations. But we do complete other population surveys in the state, one of which is our brood survey that we do every year from June 15th to August 31st. This is basically a passive way of sampling, but it gives us a decent index of also the population growth as well as reproductive efforts for that year. And these are strictly, you know, whether it's AGFC employees, natural heritage employees, Forest Service, and even in the last couple of years, we've actually stepped up the amount of public input that is provided towards these surveys as well. But this, I think, gives us more of an idea because it is more so tied to WMA management or Forest Service management, because those folks are putting down these observations as they are doing their normal, you know, daily activities, normal management. So it's on a management area. And as you can see in the last probably 3 years or so, that top graph shows the number of adults that were observed. So that's increasing, as well as you can see a decent increase in the number of chicks per adult that were observed over that that same time period. So we are seeing I think a population upswing. So that's good news in my opinion. Also with that brood survey, we're able to, folks are reporting broods and individual, you know, chicks within those broods, and they're able to take one of our, aging guide and be able to, I'm able to backdate based off of what folks report on that particular survey. And over the last 18 or so years, our data is showing that according to the observations, our peak as far as hatch is around that June 15th to June 30th timeframe. That's when the most hatches occur. Although, again, our survey sampling only goes to August 31st. So it's essentially, it may be missing some of those later hatches that are typical of quail in general. I know one study in Missouri, for instance, in Southwest Missouri that's recently conducted, they actually showed a second peak there around in the middle of August. So in order to combat these declines within the state that have occurred over 50 or more years, we have set out to demonstrate habitat management practices on select WMAs. We have 6 in the state that we classify as focal areas. And then we also have another partner agency property in Pea Ridge National Military Park that we intensively manage for quail. Within these 6 WMAs, those encompass about 11,591 acres, plus about 4,300 acres on Pea Ridge National Military Park. So over the last fiscal year, FY20, our agency habitat accomplishments, and what I mean by that is habitat improvement, cedar removal, disking, mulching, prescribed burning, WSI, all those types of practices. Even commercial harvests are included in this as well. And within those 7 focal areas, we conducted 7,626 acres of those types of habitat practices or right at 95 miles of fire break maintenance in order to facilitate some of that management. And then also within the state, I mean, our quail habitat management is not just, you know, isolated to focal areas. A lot of WMAs that we haven't necessarily just classified as a focal area, are still undergoing some of the same management, and over 35,553 acres of habitat work that would be more conducive to benefit quail, and especially on more upland sites. Obviously, we don't count bottomland, waterfowl, type habitat that is occurring, but also 92 miles of fire break maintenance on those non-focal areas. And those are the numbers in the parentheses are the number of WMAs that we're incorporating in that number. So as we do habitat work, we want to be able to see what our progress is. We conduct spring focal area bobwhite point counts. We also, as a part of these, we not only listen for bobwhites, but a suite of grassland, woodland, or open woodland, and scrub type habitat type birds that we're listening for. And for the most part, I know this is a little busy of a slide or of a table, but I wanted to be able to get all the focal areas within this chart. And you can see for the most part, there is a couple that are lagging behind and haven't really shown the results that we want. But for the most part, the rest of the focal areas are showing increases with some, some variability. As I said, quail are a species that you're not always going to see, these straight continuous growth of the population. You're going to have some ups and downs. And that was with the number of quail that were heard on those focal areas. But also, again, as I said, these are charts depicting the early successional species that we're also monitoring for. You can see some good increases at Harold Alexander for indigo buntings and field sparrows, prairie warblers, painted buntings, and even yellow-breasted chats. Kind of see very similar results in stone prairie. Again, field sparrows, indigo buntings, and chats all are increasing. Mud Creek, same thing. That Mud Creek's up in the northeast part of the state there. So again, the numbers in parentheses next to the name of the WMA is the number of points that we run, and those number of points are obviously based on the amount of acreage that we were classifying as focal area. Robert L. Hankins Mud Creek is around 1,000 acres. Stone Prairie is around that same, about 980. And then Harold Alexander is about 3,500 that we're classifying as quail focal area. Little Bayou down in the southeast part of the state, it's about 1,200 acres. And you can see very similar. We're seeing some increases overall in these species that I discussed. Even some cool, some pretty neat species that require even more larger tracts of this in the grasshopper sparrow. And let's see, Jack Mountain, very same results, all for the most part increases that we're experiencing in our monitoring. And I think that's due to kind of the intense habitat work that's occurring. Pea Ridge, very similar, a little bit of fluctuation from year to year, but still, I think, stable to increasing as well. And Hope Upland down below. So in addition to spring call counts or point counts, we also monitor the fall covey populations on these focal areas. And you can see again, for the most part, you can see increases in most all the areas other than those two, Mud Creek and Hope Upland, just aren't quite responding like we would like. But I think some of that is maybe lower populations, if any, to be honest with you. And Hope, I have yet to see much of a gain there at all. So as we progress through our management, we are working with University of Arkansas to develop a habitat suitability index for the state. We want to be able to not only have focal areas, but we want to focus in on what I call focal landscapes. And we're going to take this habitat suitability index as well as our call count data, and then also include data from NRCS and other cost share data. And what I mean by cost share data is basically where folks are spending cost share, where there is essentially landowner interest in doing more habitat work. And we're going to create some areas throughout the state, larger, thousands of acre areas where we can focus our attention, and then thus also put some call count routes or breeding bird survey routes to over time monitor the change in the populations of quail and those songbirds to hopefully correlate that with the gaining interest and gaining support and implementation of habitat within those landscapes. Because that's truly what's going to overall affect our populations of quail, is by doing a more landscape scale effort. So I talked about what we're doing on our WMAs, but we also have, over the last 3 or 4 years, we've reached out to partners through various avenues, whether it's requests for proposals to do different projects, or just simply provide maybe some type of herbicide or something like that to do habitat work on not only our WMAs, but also Forest Service, Nature Conservancy, Henderson State property that they kind of rent from the Corps of Engineers, and then Arkansas Natural Heritage Commission. So on Forest Service properties, we have worked on 4 different districts, and you can see the overall acreages and what we have accomplished on there through providing some monetary funds there towards these different habitat efforts. In total, since 2017, 1,200, a little over 1,200 acres of habitat work has occurred on these ranger districts. On Heritage Commission properties at Warren Prairie, we have, and I think this actually has increased since the last time I gave this presentation, but at least over 380 acres of TSI were accomplished on Warren Prairie. Plus, we, through the Quail program, we funded 2 years worth of a position for a native seed coordinator position to help get that program off and running, to get a plan, and then to start the implementation of getting essentially a native seed industry in the state, and help progress that so that more seed, local ecotype seed, will be available in the future for us to do more habitat restoration. With the Nature Conservancy, we were able to do a little over 2,600 acres of prescribed burning, TSI, and invasive control. And then at the Henderson State property, about 189 acres of privet control and timber thinning. So overall, with all those together, it's about 3,214 acres. One of our very close partners that we work with, in fact, again, one of the areas that the Park Service owns, Pea Ridge National Military Park, is one of our focal areas. And over the last 4 years, we've completed about 4,333 acres of habitat work there through providing funding to them to do that. So a big part of what our goal is to restore landscape scale habitat. And in order to do that, we must, absolutely, it's a must that we work with private landowners, because around 85%, 87%, I don't know that anybody has that quite narrowed down exactly what percentage private land is in Arkansas. I tend to see it vary, but I do know it's quite a large amount. And so we have applied for and received a RCPP grant through NRCS to help provide additional funds besides just the normal EQIP and CSP and some of those that are just standard every year practices that have fund pools. We applied for this and got about $600,000 for over a 3-year period. And currently we're in the second year, we're going on the third year of this program, and we have funded 12 contracts and a little over 1,100 acres of habitat work through that. We've spent about $372,932 NRCS dollars, and then as a part of this, to help make it more enticing, Game and Fish has put in the other NRCS pays 75% of the cost of the practice, and Game and Fish pays the other 25%. So that amounts to about $100,000 on our part. And the intent is to, with these, we focused a lot of our ranking criteria to reach out to those landowners that are close to our focal areas within that North Arkansas area. And one of our focal areas in particular is Harold Alexander in Sharp County. And these dots on here represent projects that have been contracted through either this RCPP program or our Working Lands for Wildlife initiative, or even EQIP, for instance. And so you can see how it's slowly building upon our efforts that we do on the WMA. So we realized right off the bat that when we started this, that this kind of quail push, that, you know, at that time we only had 10 private lands biologists within the state. So that essentially equated to a biologist per 7.5 counties. And so we thought, man, that's not going to be enough to keep up with, you know, what the demand is for technical assistance. So we reached out to NRCS and Quail Forever, and were able to get a grant that allowed us to match funds with NRCS. And we hired 8 additional farm bill biologists to help facilitate all that work that would be coming in. And over this last, I think it was when we started this was 2017 to 18 timeframe, those QF biologists have been able to contract / complete over 69,000 acres of habitat, or at least contracted or completed that many acres within that time frame. Also, as a part of this, we want to not only get contracts out there, but start bringing more awareness to the need for habitat. So these folks have reached out to the public through workshops, and in fact they've done over around 275 during that same time period. And at those workshops there was a little over 7,000 attendees. These QF biologists have also completed over 2,000 site visits to varying landowners throughout the state. And you can just see over to the right hand side, that's kind of their what they've done up to this point, all the landowners that they've visited with. So in addition to having farm bill biologists, or essentially another set of private lands biologists, we have a 5-member crew that goes around and does primarily prescribed burning on landowners' properties. Also helping with some public lands as well as needed. But this crew also does TSI on smaller acreages and things like that. They do spraying, invasive, Sericea and fescue and Bermuda type sprayings. But they are the ones that are physically going out and implementing a lot of these plans that the private lands biologists or the QF biologists are writing. So they are, to folks that don't actually have some type of a cost share, the work is free. Now, if you do have a cost share through a NRCS program, we do ask that you pay whatever it is that they're completing for you, whether it's the burn payment or TSI payment or whatever that at that point would go towards the crews and knocking that amount off of what Game and Fish pays for this type of crew. So all the staff have been hired since May the 4th. We've had a little bit of turnover, but we're right back to the full 5-member crew, and they have completed up to this point about 5,800 acres of habitat work during that time period. I've talked about this in, I think, another Audubon presentation, but we also realized the need to bring awareness for native forages as a viable option out there, because folks I think there's not as much promotion of native grasses as a true forage source. And so Game and Fish has developed this program, and it's where we provide the seed, the herbicide, even a cover crop if we need to do additional sprayings, and then also a deferment payment during the time frame in which the grasses are establishing. So we pay a $65 per acre deferment there. We have actually four people that have signed up so far. That has equated to about a little over 140 acres that people have signed up. It's from fields that were as small as 15 acres to as large as 75 acres. And we do actually have two other ones. The other one in White County down there that I said was interested, he just now signed up. He's going to plant about 26 acres in this, but there's a couple other ones that are interested in Drew County that's just trying to make up their mind about this. But again, just to try to present this as something that is out there that's viable, not only as a forage source, but obviously as a more wildlife friendly option to cattle producers. And then, of course, we have to bring further awareness to our cause for the quail, for quail habitat, and to show, provide awareness. We have implemented a Northern Bobwhite conservation stamp. The very first one came out in 2018, and we are fixing to roll out the 3rd edition this year. Currently, we're in a partnership with the Arkansas Game and Fish Foundation and Cane Hill Foundation up in Northwest Arkansas. They have hired the artist for this year's rendition, and it's going really, really well. The artwork looks really good. Currently, we have prints available, though, of the last year's quail stamp, which you see here to the right. They are $36 for unframed and $54 for framed. You can get that through the foundation. But also, I encourage you, if anybody ever asks the question of, well, what can I do? Maybe I don't necessarily have property. Maybe I don't necessarily hunt. But if you do want to see some change in the bobwhite habitat restoration, this can certainly provide a revenue source to help implement more of that. Previous revenue from the first sale of the stamp was $23,000. Currently, we have produced about $38,767. So what we did with the first amount that you saw there is we implemented a project on Fort Chaffee. We feel like that is the kind of our shining star as far as the quail populations and having higher numbers. So we picked out a location on the base there, or the training center, and you can see on the top picture, you can see all the cedar that was in the area just south of that road. And after we did the mulching, we did about 216 acres worth of mulching. You can see how it's really opened up a lot of the area there within the slide to the bottom right there. We actually got some help from Department of Defense. They have a mulcher that they own, and they pitched in and helped with some of the work there. But it was a little over 6 months worth of rental, and we spent about $45,000 do the work. Here's some of the results, the before and after shots. These areas had just gone too far, too long without adequate prescribed burns. And so sometimes you have to go in mechanically and knock it back down in order to get more of those grasses and forbs and so forth to come back. And I will end with, I encourage you guys not only to buy a quail stamp, but also participate in our quail population survey app. As a part of that, during the June to August timeframe, we ask that you provide, observations of quail and their chicks, but also we've expanded this to year round. So I encourage you to help. This helps us to get a better idea of distribution, of reproduction, and kind of an overall picture of where quail are. And hopefully we can even use some of this data to maybe help with our density estimates within the state over time as we get more and more data. But as I talked about, it's mostly currently right now, it's mostly staff, AGFC staff, not NRCS, but Forest Service, Heritage Commission, and Fish and Wildlife Service. But we definitely would love for more of the public to provide input into this observation app. You can use this on your cell phone. It's very easy. You just punch in a little bit of data, punch in your location where you heard or saw the quail, or if you can, if you're out and you don't have that, you can come back to your house or office or something and input the data in a computer as well. But it's really just any time that you're outside, we would love for you to provide those observations, and the website there below will get you to where you can download that survey app on your phone or on your computer. So again, I encourage you to participate, please. We need all the data we can get to help out to make management decisions in the state. So with that, I am done with my talk.
Okay, thank you, Marcus. I've already got a question popped up in the chat. Somebody wants to know where they can buy the stamp. She says, I'm from LA. Does that matter?
No, not at all. Any of our, you can buy it online. And then if you're obviously in near a regional office or a nature center here in the state, you can go in and purchase that there. But again, you can purchase it, and you can even call one of our regional offices or the main office, and I'm sure they can sell it to you over the phone as well.
Great. Another question. This is from Deborah. She wants to know, are you also incorporating eBird data in your quail population survey?
I have not, but that is a good idea. It's a very good idea. I think we could be able to combine a lot of this data to help paint a bigger picture there, or a more conclusive picture.
And Jim asks, what happened to the quail population at Mud Creek?
What happened to it?
Yeah.
It really hasn't ever kicked off. I think one year, the very first year, we heard a couple on that. I know I heard two during the spring. And there was another biologist that was helping that I think he heard one in the fall. Yeah, ever since we started the management, maybe the management has been a little intensive maybe, and it has maybe pushed some of them off the area, because we have been doing a lot of mulching, a lot of burning over the last 2 or 3 years, just trying to get it for a crude term, whip back into shape, because it had grown up tremendously. A lot of the fields were basically this 15, 20 foot tall sweet gum, box elder type stuff just growing in there, and it was just a kind of a mess, so to speak.
Interesting. Deborah asks, are quail stocked in Arkansas?
No, they are not. Pen-reared birds or domesticated birds do not work. There's about every agency within the range of that has quail has tried that over, back in the 50s and 60s, and that was shut down. There's been a tremendous amount of research on that because, I mean, let's face it, that's an easy way to, kind of see more quail, I guess you could say. But it's just a failing, a fruitful effort. There is some research over in tall timbers where they are moving wild bobwhites and showing some success. I don't necessarily, I think if you guys still see my screen, we still have fair to decent populations of quail that we can build off of if we just work on getting these larger tracts of habitat back into shape. So I don't see a need for it right now.
I live near the Buffalo River at a place called Irby, and we used to have quail there. I don't see a major change in the habitat, but now every few years we hear what we think is one lone bird calling. Is there any other reason why habitat that looked good is no longer viable for them? Is just the population too low?
Well, I think you may have possibly had a smaller population, and looking at maybe your area right within your house there or whatever, and maybe some of that hasn't changed, although I do get that from a lot of folks. They're like, oh, nothing's changed. But just like when people see their their grandkids, for instance, and they haven't seen them in a long time, and how they've grown, you don't if you're around it all the time, you don't so much see the changes. I personally think that have occurred. I can definitely show you, like, for instance, I could show you Pea Ridge. It went from in the 1940s to a very, very open area. And here in 2016, whenever I had one of the later aerial photos, it was just completely covered in cedar. And obviously there had been a lot of non-native grasses established, just areas grow up. But yeah, it takes a landscape to really hold a population of quail for a long time frame. And so if a lot of that landscape has changed over time and not, it's no longer in good, suitable habitat, then, it's just over time, it's going to slowly dwindle the population because they just simply don't have enough areas to subside.
Well, thank you so much for the presentation. Very interesting. I'm a little bit encouraged to see the efforts that are being made to bring back the quail. So thank you so much.
You bet.
All right. Next up is Christine Gurley. She's studying painted bunting breeding ecology for her master's degree at the University of North Texas. She has a BA in biology from Trinity University. Very interesting background, and things that she has done between that time of getting the BA and getting the master's. I wish there were time to share a little bit more of that with you, but I was intrigued with the volunteer work she's been doing in environmental outreach for underprivileged children. But there are many, many other things I'd like to share with you about Christine's background. But since we're a little pressed for time, I guess I'm going to ask Christine if you would take over and tell us about the research on the painted bunting breeding ecology.
All right, thank you. All right, can you guys see that all right?
Yes.
There we go. Okay, so hello everyone. Again, my name is Christine Gurley, and I am a member of the Painted Bunting Research Group at the University of North Texas. And I'm going to be talking about some of my master's research and also some of the research of the entire group. We are located in the Dallas-Fort Worth area of North Central Texas. So we have painted buntings that are part of the same larger breeding population, as if you're in Arkansas, we are a little far from you, but we're in the same section of the painted bunting population. Let's see. Oh, my screen disagrees with me. There we go. Before I get into it, I wanted to make sure I thanked my collaborators, the members of our team, and our funding sources. And the Arkansas Audubon Society Trust has provided funding for us for several different projects in the past few years. So you guys have made this research possible, and we appreciate you. And of course, this would not be possible without the entire members of the team. This is not everybody. This is only some of us. It takes a lot of time and labor to do wildlife research. So big thanks to all of my colleagues. All right, so painted buntings, they're very popular, of course. It's fun to see them, but why would researchers be interested in studying painted buntings besides that they are cool? So our research group is interested in this species for two general reasons. And the questions I'll be referring to fall into sometimes one and sometimes both of these categories. So our two general reasons are first to understand and protect this one species. Even though painted buntings are fairly popular with the public, there is a lot of relatively basic biology that is not known about the species, especially in the western half of their range. That's the Texas, Kansas, Oklahoma, Arkansas, that section of the population has not been studied as well as the eastern half of the United States. So we want to know more about this species to help with conservation and better basic knowledge. And we can also use the painted bunting as a case study to understand patterns in songbird behavior and evolution that apply to other species as well. So what do I mean by these larger patterns? I'm going to introduce a few ideas. You may have heard these before. So one big idea is what is up with color in songbirds. And on this slide, I have some examples of 4 different species of songbirds that you may be familiar with. I have pairs of pictures. The male is labeled with an M and the female is labeled with an F. And the males, as you see here in these examples, are brightly colored or may have dramatic contrasting patterns. Look at the black and bright orange on that red start. And the females are relatively camouflaged. Some of them are very, very difficult to see when you're out in the field. And the explanation that you may have heard before is, well, the males are bright colors so that they can show off to the females, and the females are camouflaged, so if they're sitting on a nest or feeding babies, they're not in as much danger from predators. And this is accurate, but it's also a little bit of a simplification. Have you ever wondered, okay, why does this bright color actually work? We're used to seeing really colorful birds, but what is the point of that bright color, since not all species have bright colors? Why aren't they all just brown? Surely it would be safer to be brown, like that female, instead of being bright blue or bright red. You're pretty obvious to predators. And so the general explanation we have for these species, including the painted bunting, is that color is communicating from one individual to other members of his species. A brightly colored male is showing off something we researchers call quality. And that means a bright colored male, as opposed to a less colorful male in the same species. That brighter male is probably healthier, he has better genes, he is likely to be able to defend a territory with better food access in it, and it turns out to be very difficult for them to lie with the color. They're often not physically able to grow bright feathers unless they are very healthy. So that bright color is a message from the males to the females and to their rivals about, hi, I'm healthy, I have good genes. If you are a rival, you should stay out of my way. And that male is telling the females that he is a good potential mate. And this message is really urgent for these birds. Many songbirds don't have very long lives, less than 10 years. If that, 10 years would be pretty good for painted bunting. And they also have a really high risk even if they are able to get a mate, many of their babies won't survive to adulthood. For birds that have an open cup nest, that's kind of, if you picture a bird nest, that you're picturing an open cup nest, probably. 80 to 90% of the babies in those nests will not survive to fledging. And so then that 10 to 20% that makes it still has to survive migrating before they're part of the next generation. So it's really urgent for these male birds to communicate to the females that they are good quality mates. They're telling her they're fabulous. So you would think with that urgency and with that time limit, they would develop their bright colors as soon as they're physically able. And most species where the male's colorful, that's what happens. The males develop bright colored feathers when they're very young, in their first year of life, so that when they're old enough to reproduce, they already have that message prepared and they can start trying to reproduce as soon as possible. So then here's where painted buntings are a little weird. Instead of doing this urgent message and showing those bright colors, painted buntings do this thing called delayed plumage maturation. Delayed plumage maturation means when a bird is old enough to be reproducing, they do not get their complete adult plumage until sometime after that first year. They're old enough to reproduce. You would think they would need that adult plumage to show off, but they don't develop all of it. Most species, if they have that delay, you can at least see some patches of adult plumage. But in painted buntings, they don't get any adult plumage, even though they're old enough to reproduce, until after the first full summer for the males, where they're old enough. So on the left here, I have pictures of an adult male. He's colorful. On the right, I have pictures of an adult female. She's camouflaged. And in the middle here, I have a young male. And that male looks pretty much like a female, as far as we can tell. And this is very strange that he looks so similar to a female. No patches of adult color at all. And every single painted bunting will do this. Every male started out and spent his first summer, where he was old enough to breed, with this very cryptic female-like plumage. And so our research group wants to know, what is the deal with these green males? Why do subadult males have delayed plumage maturation? Why do they look like this? And what difference does it make to the behavior dynamics of the rest of the species? It could change a lot of things if normally you have territorial males who are colorful and females, but then you also have these other green birds that sort of look like females, but they're not. So we wanted to know, why did they evolve this way? And what difference does it make to the rest of the species? So where we do our research, again, we are in the Dallas-Fort Worth area. On the map, I have a red star about where we're located, and that orange shows the breeding range, the western half of the painted bunting breeding range. And from the pictures, you can see we're in this combination of prairie and this matrix of shrub and trees. The first thing we asked was, okay, in our specific study area, we want to know who we have to work with, what birds are there, and what the population turnover is. We are working in about 100-acre space within a wildlife restoration area that's about 3,000 acres. And to track the population and which painted buntings are present so we can understand their behavior, we tracked their territories. So this map with the circles on it, each circle represents the singing territory of one male bird. This is the data from 2 years ago. And then here's the data from last summer. Again, each circle shows where one territorial male lives. And you can see that we have quite a lot of birds on this space. On average, we have about 47 territorial males within this 100-acre space every year. And to get a better understanding of how these birds use space, one thing we wondered was, if the young males are green and they look different, do they also have different territorial behavior? Do they go different places? Do they interact with the colorful males the same way? To get a better idea, we have an ongoing project where we put little bitty radio transmitters on their back. They're rather difficult to see, but there's a long antenna sticking out from the bottom there. And this is still very much in progress, but here is some preliminary data. For the young green males, we thought, well, if they are green so that they look like females, or to show that they are younger and less of a threat than the older colorful males, we would expect them to move farther and to not have as strict of a territory as the colorful males do. And so our preliminary data we found seemed to support that. We had a really difficult time tracking these green subadult males. They travel widely and often stay, we think they're staying close to the ground because we lose radio signal from the transmitters pretty quickly. But they do seem to overlap with the territories of the older males. Each box here is a different individual bird. And the adult males behaved pretty much the way we expected. They have a territory that they spend most of their time there, and they don't overlap much. They're very aggressive. So in the future, we're going to expand this data set to double check and have more information about where the younger males go, because they certainly do not appear to be maintaining territories the way the older males do. But we don't know where they're going or how far. And we also don't have a clear idea of where the females go. So one of the projects that is ongoing is to expand this radio tracking data. Another question that we asked is about multiple paternity. That means in a nest, does a female have just one mate and all the babies in one nest are from that male? Or does she mate with multiple males? And those green males, do they reproduce at all? They look like they should be able to, but they also don't have the bright colors we would expect. So we wanted to know what these guys were doing. To do this, to find out, we collected genetic information about groups of babies and as many of the resident adults as we were able. In an ideal situation, we would have genetic information from the mother, the social male or the male with a territory near where that nest is, and other males in the area that could have been fathers of the nest. And this data is really difficult to collect, but even though we had a fairly small sample size, we're really pleased that we were able to document multiple paternity. This is one of 2 nests where we have genetic confirmation, the first time for this species, showing confirmed that they do have multiple males fathering babies within the same nest. And we think the multiple paternity rate may be fairly high in this population, since we had about 20% of the nests had multiple males. That was more than we expected to find. So that will tell us some interesting things about the genetic diversity of this population. And I wanted to point out here real quick, that's a cowbird. So we also documented, as far as we know, the first time that anyone has confirmed in our region that brown-headed cowbirds are parasitizing painted bunting nests. We're not necessarily shocked, but we did confirm that. Right. Another experiment that we're really excited about the results of is this territory intruder experiment. We thought, okay, if we have these green males that they don't look like the older males, how does that affect the territorial behavior? Older males the colorful males are very aggressive, but if they're faced with a green bird, do they even know that this is another male, or are they unable to tell? To test this, we captured birds and put them temporarily in a cage in the territory of another male and watched to see what the territorial male would do about these birds in their space. And what we found was that a colorful male reacted very aggressively when we put another colorful male in his territory, but less aggressively if we put a green male in their territory. In fact, they tried to attack other adult males sometimes. They would swoop at the cage, but they never tried to attack younger males. They would sing at them and come closer to investigate. But we did not observe as much aggression, which suggests to us that they may not be able to tell whether that green bird is a male or a female. And the most interesting, I'm going to skip this slide here. One of the most exciting observations we made during these experiments was that occasionally an adult male would display toward this green bird, which was actually another male, as if he was giving a mating display to a female, including this behavior where they bow and show off that red patch on their back. So we weren't able to confirm for sure that they thought that bird was a female in every case, but we did show that the older males are less aggressive to the younger green males, and that at least in a few cases, it seems they are not certain whether that green male is actually male. A different side of that question, can other birds tell when a green bird is male or female? We also realized that birds can see in the ultraviolet spectrum. So it's possible that a green male has this patch of color in the UV that other birds can see that humans cannot. So we wanted to check if there was dimorphism in the UV spectrum. And to do this, we collected feather patches from several different parts of the bodies of young males, females, and adult males. And we picked these spots based on where the colors are bright in the older males. And we thought maybe there's UV coloration on those same spots that we can't see on the younger males. And we are still working with this data, so we can't say for sure, but our data so far looks like females and subadult males are similar in the UV spectrum. So I say they might be similar because we're still working on the strict statistical definition of whether a color is different or not, but we think that they're similar. So that combination of this ultraviolet data with the feathers and our behavioral data, where the young males are treated less aggressively, and our territory data, where the green males move around and use space pretty differently from the older males, suggests to us that these young green males do not develop that adult plumage for a specific reason, that it is an adaptive strategy to allow them possibly to mimic females. And we're really excited about that potential. We were hoping to document that. And so we're going to keep working with the data and hope that we can get even clearer results with these territorial experiments. I'm going to skip that slide as well. So our ongoing research with these birds is that territory intruder experiment. We had really cool results, and to get a clearer idea of whether those green males were being treated like females or just as less threatening than an older colorful male, we want to expand that experiment and add female birds and put female birds in cages as well, so that we have a reference point for what a female bird is treated like. We didn't do that before because we were having trouble catching the females, and we finally figured out how to catch them consistently. So we're hoping to do that in the future. We're going to keep working with that feather data and confirm that whether dimorphism is present or not. Again, we think that the green males and the females are the same, but we want to be extra sure. The subadults, we have genetic data about multiple different nests, but we haven't been able to confirm yet whether the young males actually reproduce or not. So we're going to keep working with the genetic data and see if we can identify any babies that were fathered for sure by a male we know was green, a subadult at that time. And finally, we're going to keep adding to our information on their population turnover and hopefully get some exciting data about where the females go all day and what their movement patterns are, and compare the female movement patterns to young males and adult males to give us a really good detailed picture of not only the breeding behavior dynamics, but also the spatial use of this species. All right. So that is the end of my presentation. I was afraid of running out of time, so here we are.
Thank you so much, Christine. Fascinating research. We recorded all of this, and I'm gonna have to go back and listen to some of that again. It's really fascinating and so well presented. I'm gonna ask any of you, if you have questions, please put them in the chat because we are, due to technical problems, we are running a little bit late. So if we don't get to all of them, we'll try to get the answers to you. Deborah asked, how do you know the gender of the green birds you catch?
That's an excellent question. The short answer is with difficulty and practice. So there are several reference points we can use. One is that when you catch a bird during the breeding season, most of the male birds have this external physical sign on their body. Birds do not have external genitalia, but they have something sort of similar that you can see on the bird when you have it in your hand in most cases. So if we catch a green bird that has that, then we're pretty sure that it's a male. And we also have a trick to aging them, that if we get a green bird that has a certain pattern on its wing feathers, that's common in a lot of different bird species. There's a specific pattern that only the adult females have. So if we get a bird that has that, we know that it's female for sure. We do, however, get some birds that we just cannot tell. They don't have clear physical signs, and we have to mark them down as unknown. We might eventually sex them in the lab if they're really genetically important.
Well, thank you. I have a feeling that hope you won't forget us, because I suspect we might want to have you back in a few years to catch us up on all what you've discovered. There's a comment here I'd like to make sure you're aware of. It's from Rosalie, and she says, thanks, that was so interesting.
Thank you.
All right, we are going to move along, and our next presenter is Caitlin de Bellis. She has a bachelor's degree in environmental science at Paul Smith College in New York and made her way to Arkansas, where she's currently working on a master's at Arkansas State University at the Rowland Lab. She's particularly interested in changing morphology of North American birds as a response to climate change. Caitlin, are you there and ready?
I am ready.
Great, thanks.
Can we all see my screen?
Yes.
All right, thank you. So thank you, Jack, for that wonderful introduction. As you said, my name's Caitlin, and today I'm going to be presenting to you guys a little bit about the Bluebird Trail in Jonesboro, Arkansas, and how this relates to Bergmann's rule, as well as some other general happenings with the Bluebird Trail.
Hey, Caitlin, you're sharing your desktop and not your PowerPoint.
That's so strange. Hold on. Okay, are we better now?
That's it.
That's it.
All right, plowing ahead then. So there are two main concepts that I want to bring to your attention as regards to this. So first is Bergmann's rule, which is a biogeographic rule explaining surface area to volume ratios, or basically just how an individual can control its temperature. So in warmer climates, you're typically going to have smaller individuals because you don't want all that baggage. In cooler climates, you're usually going to have larger individuals because, you know, in the winter, you want to have a nice puffy jacket. You want to be nice and big so you can stay warm. Next is climate change, which I'm sure we're all pretty familiar with, as it impacts most, if not all, ecosystems. And since 1880, we've seen an increase in the average global temperature, as well as the average precipitation, specifically in the US. However, as we are probably aware, we're seeing a lot of droughts in the southwestern portion. And then lastly, we're seeing an increase in the extreme weather events. Now, as climate change as it pertains to birds, we're seeing a lot of different alterations, first of which being phenology. We're seeing that laying dates are moving earlier in the spring, and then migration is also being impacted, where a lot of migratory species are facing mismatch, meaning that they're either arriving earlier or later to the breeding grounds, which impacts their success in the breeding season. We're also seeing changes in distribution. So as we can see in this really cool graphic of tree swallows, their wintering and summer ranges are moving more northward. However, it doesn't just stop there. We're also seeing changes in their distribution in terms of elevation, not only with tree swallows, but with other species where they're moving farther up in elevation. Next, there's population changes. And as we can see from this graphic, there have been basically declines in all bird populations in most breeding habitats, except for wetlands. And then lastly, something that I'm interested in is the morphology, or basically the sizes of birds. And as a recent study that came out in 2019 has shown, we're seeing that there's a general increase in wing chord, whereas in tarsus and mass is decreasing. And this is particularly with songbirds in North America. So with Bergmann's rule, climate change, and bluebirds, we're seeing the rising temperatures. And then with Bergmann's rule, where we're having smaller individuals with warmer climates, we're expecting to see that bluebirds are decreasing over the past almost 2 decades. So a little bit about eastern bluebirds, even though I'm sure we know so much about them. They are a common, widespread songbird species in eastern North America. They can be residents or migratory, resident meaning that they don't migrate during the winter. They stay throughout the winter. They are cavity nesters, and they typically have 1 to 3 broods per breeding season. And then within the brood, they have 1 to 6 eggs. However, something interesting that we noted in 2016 and in 2020, we saw that 3 pairs had 4 broods each, and we're actually working to get this information published. So that's pretty cool. So a little bit about the Bluebird Trail. It started in 2003. We have about 150 boxes that we monitor throughout the breeding season, which is March through September generally. And then we also band and take data on the fledglings and the adults. And then we have Annika releasing a male bluebird, which is cool. So what do we actually do? We monitor the nests, meaning we look at the progression. So when the nest first starts, when it's complete, when the first egg is laid, when the eggs start incubating, hatch, and then when the fledglings actually leave the nest. However, not only bluebirds use these boxes. It's not like they're only for them. So we do have other species like Carolina chickadees and Carolina wrens, as well as this particularly angry tufted titmouse that was pretty aggressive with me, but she was a real good mama. So once the eggs hatch, we try to trap the adults. And when we do catch those adults, we sex and age them. We collect their mass, wing chord, and tail length. And then if we previously haven't caught them, then we give them a USGS band, as well as 3 color bands to give them a unique combination. That way we know who they are. And then similarly with the fledglings, we basically just grab them out of the nest because they don't move much. And we take the same measurements, sex and age, mass, wing chord, tail length, and then we also give them the USGS band. However, we only give them 1 color band, just because their legs aren't long enough to give them 3. So if we do recapture them the next breeding season, we will give them the full color combination. So once we take all those measurements, I take them into the lab and I run some models. So basically I'm looking to see if year definitely has an effect on wing chord or mass. And I use adults and fledglings since we have data on both of those. And I turn that into an age classification called ADIO, which I just gave it that name. So all the adult birds, which would be AHY, SY, ASY, are going to be adults, whereas HY birds, or basically fledglings, are going to be young birds. The sex is just female or male. If we couldn't determine it for whatever reason, especially with the fledglings, those won't be included. And then lastly, I include year in the models. So basically we're just looking to see if year definitely is in the best model. So drum roll, are bluebirds shrinking? No. But this may be for several different reasons. So though you may see that my sample sizes are relatively large, they do only span for 18 years, technically 17 because we missed out on 2011 data. So we might not have the temporal breadth that we need to detect a trend. Also, there's this anomaly called the warming hole, where Arkansas is part of that, and we're not seeing the warming trends that the rest of the country is noticing. So if there's no difference in temperature, then there might not be a difference in morphology. It's just not there. So hopefully if we collect more long-term research, we'll get more data, so we may be able to detect a trend later on. So since there were no detected trends, Bergmann's rule obviously isn't applicable, but only with this population. Obviously, we can't make statements for the rest of the bluebirds and the other portions of their range. So switching gears a little bit, I'm going to talk to you guys about the other projects that we're doing this year. We're participating in a nest parasite project with the University of Connecticut, where essentially we're just collecting nests that have at least one chick in them, only bluebirds, no other species. We record the fate, and then we Ziploc the bag, and we send it off to UConn to be analyzed by their parasite team. So that'll be cool when we get those results. And then something that is particularly interesting to me, my advisor, Dr. Virginia Rowland, started up this project, which I'm helping her out on, to see if size mattered for eastern bluebird survival of the winter storm. So if we remember and rewind to like the second week of February, I know I was pretty excited about it with all the snow and the low temperatures, but we definitely had about 9 to 15 degree difference in terms of a decrease from the normal temperatures that we would be experiencing during February. So if you see on the left, especially with the top picture, the previous week, we had about normal temperatures, but then fast forward to the next week and we decreased so quickly, which unfortunately had a pretty severe impact on the bluebird population here. So after the storm, we gathered up some people and we went to the nest boxes to see if we could find any bluebirds that unfortunately perished from the storm. And we did. We found 69 bluebirds. 43 of those individuals were banded, and then 22 of those banded individuals were fledglings from the previous year. And then if we see in this picture right here, the star represents the box where we found 16 bluebirds because they were all trying to huddle together to stay warm, which is pretty sad. And basically, all these lines are showing where the bluebirds either were parents or hatched from. And they all kind of congregated here, and they all kind of came from less than 1 kilometer away, except for one individual, which strangely came from more than 1 kilometer away. So that's kind of interesting to see where they all congregated, where they came from. So continuing, we are in the midst of the breeding season. So I've been out there trapping the fledglings and banding them while Dr. Rowland is dealing with the adults. And unfortunately, we've seen drastic differences between the previous years and this one, with about a 53% decline on the average previous years, a 96% decline from the previous years, and then a total 85%. So it's very noticeable, even to the point where like you don't need math for this. Even today, I got stopped by a resident who has like 10 boxes just on their property for fun. And he was saying that he didn't have any bluebirds, whereas the previous years, he's had like at least 7 of them occupied. So it's definitely noticeable. So we still have some time in the breeding season. I'm still finding new nests. So not all is lost. And in previous years, especially in the 70s, where winter storms were affecting bluebird populations, they did bounce back. These populations did bounce back. So it's not like all is lost. Bluebirds are never going to be around again. However, this does exemplify how significant these extreme weather patterns are affecting bird populations. So not to end on a sad note, I do have some field pictures. I am very fortunate to have some volunteers come out to hang out and check out these cool birds. So we have Jen, who's actually has a recorded presentation later on, and Alyssa from the Herp Lab. They're holding bluebirds and a chickadee respectively. And then we have some day 7 bluebirds with a nice fecal sac. We also run into horses. There's a field tech, Joe, holding a titmouse. And I have a lab mate, Monica, holding a male bluebird. And then Dr. Archie Ryan is actually rehabbing a bobcat. So that's been cool to run into her as I was checking the boxes on his property. But lastly, I would just like to acknowledge my advisor, Dr. Virginia Rowland, and my committee, Dr. Thom Bovis and Dr. Travis Marsico, A-State and the Rowland Lab, as well as my field tech, Amanda Trusty, who's helped out over several years now, the superb cohort and friends who have come out to help me out, whether for their own selfish gain of getting to hang out with cool animals or genuinely want to help me. And then lastly, my funding sources, the Arkansas Audubon Society, Arkansas Game and Fish Commission, and the Superb program. So does anybody have any questions?
I can tell you that several of the people who have put in questions, you answered them right after they put the question in, so you must be psychic.
I guess so.
Somebody asks, for the nest parasite study, what were you looking for on the collected nest?
I'm sorry?
What were you looking for in the collected nests on the nest parasite study?
So I believe they're looking to see if the nest parasites impact if the chicks survive to fledging more, like what they are looking to see if there's mealworm effects as well, but we don't use mealworms for our boxes. So there are different landlords, as they call them, participating in this. So I guess to see if there's a difference in the geography of the parasites, as well as to see if it actually impacts their survival.
I had no idea that we were living in a warming hole here in Arkansas. That's good to know. Is anyone doing this kind of study on the Bergmanns and the impact on bluebirds in other parts of the country?
Not that I'm aware of. I mean, I'm sure it could be done. But I'd be interested to see that, especially if they're outside the warming hole. Right. We'll see if it actually affects them. But I mean, I don't know if there's been such long-term studies in other places. So Great. It'd be cool to see.
That would. Thanks so much. One thing I learned from you is when you're typing Bergmann, make sure you put in both ends. Thank you.
Yes. Now you know.
Yeah, right. Michael says what I think most of us are thinking. Job well done. Keep up the good work.
Thank you.
All right. This last presenter is, I think, not able to actually be here, but she sent in a very fascinating video clip that we're going to show you. This is from Jennifer Terry. She's a master's student at the Laurie Newman Lab at ASU. I particularly like to have this presentation because it shows you that Audubon is more than just about birds. We know everything's connected. Audubon uses birds as the symbol for all of the natural world. And Jennifer is going to tell us about her work on turtles. And the title is Acute Stress Response in a Freshwater Turtle in the Arkansas Delta. And fortunately, since I can't get my video to work, Dan Scheinman and Samantha, we were able to send that over to them, and they're going to share their screen with you.
Hello, my name is Jennifer Terry, and I am a master's student at Arkansas State University. And I'm unable to join you all live today, as I likely am in the field trying to catch and sample turtles as you listen to this talk. But I hope you all are having a wonderful meeting, and I'm talking to you today about the acute stress response in a freshwater turtle in the Arkansas Delta. In the fall of 2019, I was a recipient of an Arkansas Audubon Society Trust Grant, and so I'm excited to talk to you guys today. In my presentation, I will talk about what we do, what we find, and what we do with the turtles that we catch. Then I'll just go a little bit into the types of questions I ask with this type of information. I'll share a bit about why turtles are awesome. And lastly, I'll talk about how this type of research can be applied. My field site is in Cross County near Cherry Valley at a private area known as Beritage Farms. We have a number of methods that we use to catch turtles, including hoop traps, box traps, bike nets, and just simply grabbing them when we see them on the road or on the property. Turtles may be moving somewhere to find a suitable nesting habitat, or they may be moving from one water body to another. So sometimes we just happen upon them. And we typically use canned sardines, meat or fish scraps, and sometimes even dog food to bait our nets and traps, since we are primarily catching freshwater turtles. Most of our traps are accessible by wading in, but sometimes we get in a small boat so we can trap in deeper water. And my favorite part about my research is that you never know what you're going to get until you pull up the trap several hours later. Sometimes we come up on a net in a boat and we see the whole thing shaking if we have a lot of animals or big animals in there. And so you never know what you're going to get. And it's really exciting. It's a surprise every time. And my type of research and the things like what it entails provides a really cool opportunity to share this experience with students, my peers, and other visitors who can get involved based on their comfort and their experience. And the Arkansas Delta is amazing in terms of its turtle biodiversity. We happen to be able to find all of the expected species in the general region at my field site. And while my study organism is the very abundant red-eared slider, you may recognize them by the red stripe behind their heads. They're very common throughout the state. We also catch spiny soft shells, which is the pancake-looking turtle in our top left corner, river cooters, mud and musk turtles. A mud turtle is shown here on the top right picture, the little juvenile there. And alligator snapping turtles, which is in the bottom left corner, common snapping turtles. And so it's really, really cool. And it's a really cool opportunity to trap at this property. And the opportunity of both personnel and the animal is of utmost importance to us. So we make sure everyone knows how to properly handle the different species. You don't want to be bit by any of them, but particularly with the bigger ones, the risk is obviously a little bit more if you do get bit. And so sometimes we employ household objects like a toilet plunger that you can see in the bottom picture, one to the right corner, where I am putting a plunger on a common snapping turtle. So they have a pretty strong bite and they can move pretty fast. So when we're measuring them, it's kind of optimally a 3-person team. So I'm securing the head very safely, but I'm not restraining the animal so that they can't breathe or anything, but I'm just making sure that my hands are away from the animal, but securing the animal. My lab mate, Emily, has her foot firmly on the turtle without putting her hands near the area. And then someone with constant communication can get in there with a measuring tape or calipers to measure the animal. And so, you know, we tailor each experience based on the type of animal that we catch and what needs to happen. So whenever we find a turtle, we record the time that we made contact with the animal. So that means pulling it out of the net, pulling up the net, catching the animal with our hands, because timing is really important for the blood samples and the data that we collect for my project. From blood samples, I take the plasma. So when you centrifuge or spin down the blood, it's this yellow portion that is at the top. And we use it in various assays to measure innate immune function as well as hormone concentrations. We measure each animal. So we take various measurements of the shell and we also weigh them. And each animal caught is added to our mark recapture data set. So we are able to use a drill or a file to make a mark on or hole in the outer part of the shell to permanently mark the individual. I kind of think of it as like a ear piercing in that, you know, it's not influencing really much else, and you can see it and it lasts a long time. So this way we can track the animal's growth, physiology, etc. for years to come if we ever catch that animal again. And I also use a portable ultrasound on female turtles to see what's going on in terms of reproduction. So, you know, are they developing eggs? How many clutches? So how many groups of eggs do they have developing at different developmental stages within a nesting period? And in this image, I can see a shelled egg at the top left corner here, as well as large developing follicles. And for visual purposes, imagine the follicles as growing egg yolks that just haven't had the shell around the egg yet. And lastly, we record when, where, you know, is it a male or female, things about the animal and the time that we captured them. And so I do a lot with the turtles I catch, but what am I doing with it? I'm interested in how animals manage their energy and what that looks like in terms of where energy is allocated. Every organism, including ourselves, has access to a limited amount of energy to which they can divide up to all the things an organism needs to do to survive. I like to imagine it as like a pot of gold or money, and so that every animal may spend their gold differently, to continue with this visual. And it depends on a lot of things like the animal's sex, life history, and condition. But a specific type of hormone, glucocorticoids, are thought to be mediators of this energy allocation, and they're referred to as energy mobilizing hormones. Hormone patterns also influence innate immunity. And so in my research, we use the acute stress response to understand what's going on in terms of the animal's energetic trade-offs. And so when we capture a turtle, this causes the acute stress response within the animal. And this triggers the hypothalamic pituitary adrenal axis, or HPA axis, in which a sequence of hormones from each organ are connected. From the adrenal cortex, the primary glucocorticoid in turtles, corticosterone, is released in a higher concentration than baseline levels. Corticosterone, or CORT for short, acts on other organs and processes, which cause a number of downstream effects to cope with this stressor. An example of this is that we expect to see an increase in innate immune function. And so while individuals may vary with the pure concentration of hormones or, you know, their baseline immune function, we are interested in the magnitude of change over time throughout this acute stress response, because it can provide insight to energetic trade-off dynamics and the overall state of the individual. Again, the starting point of the individual may not necessarily tell us this much. We expect an appropriate stress response to involve an increase over time and a return to baseline concentration. So this individual has this type of response And it's pretty appropriate in this example based on the smiley face. But this animal might also start at a different point, but because we see an increase and then a return to normal, that one's also considered appropriate, even though, say, the baseline may be different than the other one. However, if we don't see a change, for example, that's not necessarily appropriate, and that's indicated by the sad face. And so these are very basic examples of, what we're considering when we look at these hormone concentrations over this time period. An appropriate response in terms of immunity would be a temporary increase. So in a stressful event like a predator attack, the likelihood of injury is higher, for example. So an increase in aspects of your innate immune system or your first line of defense would be advantageous. So you want to be able to heal from any wounds that you may incur trying to survive that predator attack, for example. And so that's going to be beneficial for you. And that's why it makes sense for an acute stress response. The way that I measure this is by basically taking blood plasma in the lab and introducing it to a known amount of a microbe like E. coli. And then I provide that E. coli with nutrients to grow and incubate whatever E. coli survives from, being introduced to the immune components of the plasma of that turtle. And that allows me to compare the concentration of E. coli that remains to samples where I didn't introduce plasma and just let that amount of E. coli grow. And so turtles have an innate immune function, and so do you. So in people and other animals, your innate immune function includes your white blood cells, different functions of various white blood cells, your immune proteins, inflammation, and even when you get a fever when you're not feeling well. And this is a general first line of defense against pathogens. So it's not specialized, it's for whatever that your body may be exposed to. And so with my research, I'm finding that overall, the population of turtles I'm sampling is probably pretty healthy. While I'm observing changes throughout my field season, it doesn't seem like energetic trade-offs necessarily need to occur in the sense that animals are not giving up one crucial aspect of survival or fitness for another. So for example, we're not seeing that because they, say the females that need to produce eggs, their immune system's not completely crash, for example. But we do see general patterns. And so this is evident through the overall high innate immune function I observe. I've seen mostly appropriate increases in corticosteroid concentration. And most of the females I encountered and did ultrasounds on were reproductive. And so this indicates that, energy naturally is depleting throughout time. So we're in relation to clutch development, for example, in females, but it's not to the point where this animal is like critically unhealthy. And so we observe patterns, but it's not too crazy. And we also observe sex differences in these responses. And so it's very clear based on what we know about the life history of male and females in this population and in the species. They're doing different things at different times in terms of reproduction. And so that's kind of evident in the different responses we see between male and females. And what the work that I did in my master's for this project will inform the future work I do in my dissertation work in the same system that I'll start in the fall. And so I love turtles, but why should you? Turtles are a group that are considered one of the most imperiled groups on the planet due to anthropogenic impacts like poaching, habitat destruction or fragmentation, harvesting, road mortality, and climate change. Turtles play a number of important roles in our environment, such as predators and prey, nutrient cycling, and even providing habitat for other animals. The gopher tortoise that lives in the southeast of the United States is a really good example of this. And understanding physiology will help us understand natural changes and possible vulnerabilities, as well as understanding when things aren't necessarily responding normally. The survival of reproductive turtles So adults is crucial to the population stability because relative to the number of eggs laid, very few offspring actually make it to adulthood. And so generally turtles take a pretty long time, takes multiple years for them to reach adulthood, and then they can live 20, 30, 40, 50, even up to 100 years, depending on the species. And so they take a long time to mature, and they need a lot of reproductive opportunities for the population to remain healthy and stable. And lastly, this type of work is done in many systems, not just turtles. A quick search in the recent literature finds many physiological studies involving birds, elephants, polar bears, other reptiles like lizards, amphibians, and tons of other animals. From this information, we can learn about the similarities and differences across groups. And like I said earlier, understand the health of species and ecosystems as a whole. There are also a number of ways to understand physiology in animals outside of the methods I touched on today. For example, there are ways for you to take feathers or fecal samples and measure the hormones that are deposited there as metabolites. There are also studies in metabolism and thermal regulation and other things that I don't touch on in my project necessarily. And so I'd like to thank you for listening to my talk and your support of my project. I couldn't do this without my committee, my field and lab team, as well as my funding sources, including the Arkansas Audubon Society. So thank you, and have a great rest of your meeting.
All right, great. It was interesting. I enjoyed the clever graphics as well. A Band-Aid on a turtle. That was, and we end with something of organisms that are in decent health. So in spite of our kind of rocky start, I think we were running over here a bit, but I think it went well, and that's thanks to Marcus Asher and Christine Gurley and Caitlin Bellis and Jennifer Terry, and definitely Dan and Samantha Scheiman, who have been working in the background to keep this thing moving. I need to thank the Newton County Library. The technical problems were not due to them. They've got an excellent internet connection here. It's one of the great rural libraries in Arkansas, friendly place. So if you're up in Newton County, stop in at our library. I think you'll be impressed. You can use the same sign-in that you just did to get to this program to join the evening festivities at 7 o'clock. And the main speaker there is author Laura Erickson, who's going to be talking about 101 things you can do to help birds. And I've heard that Laura is a very interesting speaker. So go out, grab a quick bite, and come back and sign in for the final part of this. My name is Jack Stewart, and I'm the chair of the Arkansas Audubon Education Committee. And we now have other members of that committee. So next time, who knows what you'll see on our program in the fall.